transcription. Initiation. Starting the process by RNA polymerase attaching. Since DNA polymerase is an enzyme that makes DNA polymers, you can see why RNA polymerase would be an enzyme that makes RNA polymers. Elongation. Growing the strand by adding RNA nucleotides in the correct order using complementary base pairing. Termination. Ending the strand and releasing the enzyme. Both initiation and termination have markers on the DNA that denote where to start and where to stop. When you are complementary base pairing to create RNA from DNA, you must remember to use RNA nucleotides. They all have more oxygen on the sugar, so are ribose rather than deoxyribose, and every place you want to put a thymine, use a uracil instead. It complementary base pairs with adenine in place of thymine. The mRNA isn't finished at this point, though. It needs more work. There are sections that are not needed, and they must be removed. It also needs a way to attach to the ribosome and a way to protect itself from stray enzymes in the cytoplasm. It gets cut into sections while still in the nucleus and then reassembled. Pieces that are cut out and left in the nucleus are called introns. Pieces that are reassembled and become part of the mRNA will exit the nucleus and are called exons. An extra guanine nucleotide is added to the beginning, but put on upside down. This is called a guanine cap. This will help it attach to a ribosome. And a lot of adenine nucleotides are added to the end of the strand. This is called a poly-A tail. If the stray enzyme nibbles on the mRNA strand, it won't get to the information. The finished mRNA leaves the nucleus through a pore in the nuclear envelope, the double membrane. Introns stay in the nucleus. Exons exit the nucleus. Guanine cap helps bind to ribosomes. Poly-A tail, many adenines, protects against enzyme action. While RNA is single-stranded, remember that it can fold back on itself. The various bases can bond with others on the same strand. Note all the hydrogen bonds doing this in the diagram. The key sections are the ones that hold on to the amino acid and the ones that will complementary base pair with the codon on the mRNA, called an anticodon. The simplified drawing emphasizes those two areas. The tRNA brings in an amino acid to the mRNA. They attach to a ribosome in halves. If it fits, it stays until the next tRNA brings the next amino acid. Then a peptide bond forms between the amino acids with the help of an enzyme. The first tRNA leaves, and the second keeps the strand of amino acids. This continues until the polypeptide is complete. Any whose anticodon won't base pair with the codon doesn't fit and is ejected. The initiator tRNA starts in the P site. All other tRNA molecules start in the A site and translocate or move to the P site after it's vacated. The first tRNA is called the initiator tRNA. The growth of the chain is called elongation, and the finishing is called termination, the same stages we saw in transcription. You'll need to open the screen up to play the animation. Here's a view with the P site and A site. The mRNA attaches to the small subunit of the ribosome, then the large subunit attaches. The start codon is a specific sequence of bases that tells the translation process where to begin. AUG, which codes for methionine, MET, is the start codon in eukaryotic cells. See the P site and A site on the large ribosomal subunit? Here's the sequence again. Remember, these are peptide bonds being formed between the amino acid. We've looked at this before when we discussed proteins in Chapter 3, but let's look again. This is an example of how a single nucleotide mutation can be detrimental. There's a single amino acid difference between a normal hemoglobin polypeptide and one with sickle cell. A hemoglobin molecule has 4 tertiary units to make up the quaternary, 2 alpha chains and 2 beta chains. This is a mutation in the beta chain. A normal red blood cell is round and concave on both flat sides, somewhat like a lifesaver candy that doesn't go all the way through. When the multiple hemoglobins inside it are sickle cell type, they pull the round cell into a sickle shape, rather like a banana. Blood vessels can be large or small. The smallest capillaries can allow only one normal cell through at a time. So think about having a pipe just big enough to roll oranges down. You just roll them along, orange, 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 and they roll out the other end. Then instead of an orange, you drop in a banana. It's not rolling out the other end. It twists in the pipe and jams. Suddenly, you can't fit any more oranges in either. In the blood vessels, the cells downstream waiting on food and oxygen aren't getting it. And there's a crowd of cells backing up and breaking the fragile blood vessel. Clots form. Now, think about how this will feel in your thigh muscles. Not pleasant. Think about how it will be in your liver or kidney or brain. This is how it can kill. Your spleen can remove damaged blood cells, but not always quickly enough to keep from having issues. This is a serious genetic disease. I've had students in the past who have sickle cell anemia. If you have it, you may want to consider registering with the Accessibility Resource Office. If you have a flare-up, they can help you stay on track in your classes. Also, although this shows a very detrimental issue from one amino acid placed incorrectly, sometimes an error can be positive instead. Most, however, are neutral. The type of mutation shown in sickle cell is not the only one that's possible. Look at these types. Which is most likely to cause problems with the protein? The top is a normal gene. The second has had a nucleotide replaced with another. This only affects one nucleotide, therefore one amino acid. This is what we saw in sickle cell. The second has a deletion, and the last an insertion. Both of these causes a reading frame shift, as it affects every single codon after it. These types of errors have a larger effect than the substitution usually does. Why would these additions to the virus be helpful? The glycoprotein spikes will help the virus attach to the host cell, the cell it uses to reproduce. Enzymes, if it can't count on the host cell having an enzyme needed to reproduce it, it must bring that enzyme. Membranous envelope? It gets the membrane from the host cell when it exits, basically stealing part of the plasma membrane. That covering makes it able to approach the next potential host cell with ease. The host cell doesn't recognize it as a threat. It's the wolf in sheep's clothing bit on a microscopic level. The coronavirus we've been dealing with this year has both spikes and a membrane. That membrane also helps us fight it. All the hand washing and soap you've been using has been to break up that membrane and render the virus ineffective. Remember that soap is an emulsifier. It surrounds lipids and separates them. And that membrane is made of mostly phospholipids. So wash your hands frequently and well to destroy that membrane. These are the reproductive cycles of viruses. We already looked at the basic one, the lytic cycle. That's how colds work. You get exposed and the virus starts attacking your cells immediately. You start showing symptoms within a few days. But what about other viruses? What about HIV? If you get exposed, do you show symptoms immediately? No. This one can go dormant or latent. That's the lysogenic cycle. Lytic and lysogenic cycles as shown in a prokaryotic host. In the lytic cycle, the bacteriophage lands on the cell and injects its DNA, which becomes a circle. It takes over the cell, causing it to stop its normal activities and make more viral parts. Viral parts then assemble. New viruses then exit the cell and lyse it, break it, on the way out. They are now ready to infect new cells. We looked at this one at the beginning of the chapter. In the lysogenic cycle, the bacteriophage lands on the cell and injects its DNA, which then merges with the host cell DNA. It just stays there and hangs out, which is called being dormant or latent. Every time the cell undergoes division, the viral DNA gets copied along with the cell's regular DNA. At some point, there's an environmental stimulus. The DNA from the phage separates from the cell DNA, and the virus moves into the lytic phase. I remember that in the lysogenic cycle, the phage DNA lies in the genes of the host cell. That's how I keep them separate. Here's a story for you. Cold sores are caused by a virus. Once you have it for life. It mostly stays dormant in your nerve tissue, but when you're under stress and environmental stimulus, it wakes up, moves down to your mouth, and causes a painful cold sore. I don't get them. Nobody in my family gets them, except my son who caught them from his wife. Why? My mom was an absolute bulldog about them. If you had a cold sore, you had to stay away from her kids. I appreciate that now and did the same for my kids. However, our neighbor across the street, Dave, did have the cold sore virus. Dave was laying a new cement driveway. We had gone to help level it. He was stressed to the max, totally sure someone would drive on it or a dog would get in it or something. That stress woke up his cold sore virus, and it came out of dormancy and went into the lytic cycle. But this time, it messed up. Instead of traveling down his nervous tissue to his mouth, it got turned around and went into his brain. It caused a huge brain inflammation. It killed him within 24 hours. The doctors told his wife it happens in the USA about 200 times a year. Now, that number isn't statistically significant when you think of the millions of people here, but if you're someone with cold sores, it's something to think about. So if you have active cold sores, don't go kissing on people. Don't spread it. The previous examples were nice, simple bacteriophages. What about more complex viruses? And what about RNA viruses? Look at the steps in the diagram. In this virus, the glycoprotein spikes attach to the host cell, and then the membrane envelope merges with the cell membrane, acting as if it was a transport vesicle. The entire virus enters the cell. Remember, with the phage, only the DNA did. The cell immediately begins to dismantle the protein coat. The cell can always use more amino acids. But breaking up the protein coat releases the genetic material. In this case, that is RNA. The RNA begins immediately to be translated, making new protein parts that assemble and leave the cell, stealing plasma membrane on the way out. This is a lytic-type pathway. Emerging viruses are certainly in the news these days. As I write this, the world has COVID-19, a new swine flu outbreak in China, and an outbreak of Ebola, which is about under control. So where do they come from? Some are mutations of already existing viruses. This is why you get a new flu shot annually. Some are existing in one species, but then cross into another species. This is also due to a mutation, but it's detrimental for the new species. Some have been out there a long time, but just don't come into contact with their hosts. This is a current concern with global warming. Glaciers are melting, exposing all sorts of things. The potential is there for that to include viruses and even bacteria that we thought was long gone. The amount of risk is likely slight, but we should be on the lookout. HIV, or human immunodeficiency virus, is another RNA virus, but it can use the lysogenic pathway. Wait, it's got RNA. How can it merge with the cell's DNA? Those two can't merge together. This is where bringing your own enzymes comes in handy. HIV is called a retrovirus. It brings 2 strands of RNA and 2 copies of an enzyme called reverse transcriptase. Break that name down. Enzyme that reverses the process of transcription. Transcription is taking DNA and making RNA. This enzyme takes RNA and makes DNA. Once the DNA is made, it can then insert into the cell's DNA and become latent or dormant. Crazy, huh? Bacteria do not have sex, but they do share DNA. They have several methods of acquiring new DNA. Transformation is simply picking up a fragment of DNA, often from a dead or dying cell. It's as if you were walking down the street, saw a dead person, decided they had cool shoes, so you take them. The second method is having a bit of cell DNA accidentally transferred by a virus. In this case, the viral DNA got some cell DNA copied along with it, and now it's a permanent part of the virus. The last method is the closest to actual sex. It's called conjugation. Extensions called sex pili attach to another cell, draw them close, and then widen. Through the newly formed tunnel or mating bridge, the cells will give or trade copies of some or all of their DNA. This is a transmission electron micrograph of a replication bubble in humans. Replication bubble? Huh? This is a strand of DNA that's been opened up so that it can be replicated. Remember that DNA is double-stranded with the two strands held together by hydrogen bonds. This is those bonds broken in a short section of the DNA. The opening between the strands is called a replication bubble. RNA. Flipper, right? These have a specific order too. Definitions, review criteria for life from Chapter 1, they are not. Some scientists would like to change that. They did. Showed them which did, and led to a new understanding of what DNA could do. Experiment, the tagged DNA was found inside the cells. That's what's in the pellet, since the cells are heavier than the liquid. Now they knew which part was entering the cells and causing changes. Lysed. Remember hydrolytic enzymes? Same root. To lyse is to break. Lysosome. Same root. Which came first, the chicken or the egg? Are we just survival machines for our genes? Acid, ribose